I'm not trying to brag, but I do well for myself. I've always had a mind for business, but changing the world requires intelligence, hard work, and just a touch of charisma. Consider the big players, Bill Gates, Elon Musk, the common cold, all of a touch of darkness in them, a flair for the unhinged, but nevertheless we submit to their legacies. I'm HSV the second, carrying my father's namesake, herpes simplex virus, and half his industrial DNA to boot. Want to know the secrets of my success? These new viruses don't know when to quit. They lyse cells left and right. The secret is choosing when to pounce. I keep my hosts alive, strong, and good old human desire keeps me circling the globe. People think I'm just a brain, but I'm a looker. I'm a pretty big guy. My capson's a polyhedron with 20 faces known as an icosahedron. I'm all for having layers, so I sport a lipid envelope, made by host cells themselves and attached at my viral matrix. I'm a DNA guy. I rock it double-stranded and linear with as many as 84 protein-coding genes packed in. I'm a virus, so technically not alive, but who cares when you're having this much fun? I'm covered in glycoproteins, which help me bind to host cell surfaces fixed with receptors for easy entry, if you know what I mean. My envelope's made up of host cell materials gathered in a process called budding out. With a disguise like that, I'm able to enter via membrane fusion or other ways such as creating pores or fragmentation when I want to get messy. I stick with my hosts for life because I'm a loyal guy. I recruit new hosts often during asymptomatic viral shedding. With no sign that I'm running rampant, a high viral load builds up and I pass to a new property through mucus or other bodily fluids. Being HSV2, people think of me as genital herpes and my dad is oral, but we actually affect areas interchangeably. Condoms sometimes get in the way, but since I live in unprotected areas like the scrotum, butt cheeks, upper thighs, or labia, there's no surefire method. Most people don't even know I'm there. I stay away from routine STI testing because I don't cause a lot of health risks and they want to limit false positive results. It's estimated up to 80% of folks with HSV2 don't know and aren't symptomatic. But don't worry, I can still spread through them. Me and my pops, we look pretty similar and we definitely have some family traits. My genome's about half the same as his, so we have our differences. In terms of similarities, we both constrain cell destruction, except for the rare occasion that we pop out to the skin and create some lesions. Otherwise, we establish latency in the safe haven of sensory neurons and ganglia, keeping us safe from antivirals such as the cyclovir and its analogs. We usually stay in the peripheral nerves near a site of initial infection, like the sacral ganglia in the case of genital herpes. I'm most likely to come have an epithelial lice party if you're physically or emotionally stressed, so think of me as your personal therapist, gauging how you feel and giving prompt feedback when you need to slow down. Natural stress hormones like corticosteroid trigger the viral reactivation. So how does that make you feel? During latent periods, I express latency-associated transcript RNA. This interferes with natural cell death, preserving a reservoir of the virus within your cells. A protein found in the human cell nuclei called ICP4 can keep lytic expression at bay, binding up the viral DNA elements and stopping transcription. When the protein dissociates, this silencing stops, making you vulnerable to lytic phases. Transcription is pretty complex because of my large viral genome, but you should know I used host cellular enzymes to transcribe my DNA to RNA for protein and glycoprotein synthesis. Humans are finally seeing some of my construction charity work. I benefit from it too, but your nerves are my highways, so I actually have DNA coding for proteins that promote nerve repair. Most viruses thrive in acidic environments, but I like neutral weather, so a lot of antiviral defenses that neutralize the pH are just adding to my vacation. HSV can infect a lot of species, with over 100 identified strains discovered, although notably, humans are the only primate with 2 herpes simplex viruses. Genetic sequencing shows that HSV1 was infecting humans before they were human, before the evolutionary split from chimpanzees, while HSV2 jumped from a mutated form from chimpanzees to humans, and I don't want to know why. You can have HSV1, HSV2, we can have both in different regions or both in the same place. We're just not picky. So we've established a few things. I'm good at hiding in nerve cells. You never get rid of me, but I don't pose a lot of health risk. The common exception is pregnant individuals, where I can cause fetal infections or premature labor. Newborns have underdeveloped immune systems, meaning that I can potentially enter the central nervous system or vital organs, resulting in a potentially fatal infection. It's scary, but just don't be so quick to judge me. I'm a smart businessman. I mean virus. My industriousness does not need to be your demise, since chances are one day you'll be business partners with a herpes virus near you.
Ebola virus is a type of hemorrhagic fever virus that is named after the Ebola River where it was found in 1976. It is likely that humans were first exposed to Ebola by consuming an infected bat. The most widespread and fatal outbreak was in 2013 in Africa with over 28,000 cases and more than 11,000 deceased by its end in 2016. Ebola is transmitted from host to host through body fluids. It initially causes fever-like symptoms such as headaches, fever, and a sore throat. As symptoms progress, vomiting blood, having red eyes, and developing a rash is standard. It is a dangerous and deadly virus due to its ability to inhibit clotting proteins, leading to excessive bleeding. Infected people experience internal hemorrhaging that causes bleeding from the eyes, mouth, and ears in the late stages of infection. Ebola kills many people due to loss of fluids and low blood pressure. Ebola virus is a virus part of the filoviridae family. All filoviruses are risk group 4 pathogens, are transmitted to humans through animals, and have a lipid membrane and a single-stranded negative sense RNA. There are 6 species of Ebola virus, with Ebola virus, or EBOV, being the deadliest and most common and responsible for the Ebola epidemic of 2013. All members of Ebola virus can infect humans and other primates, except for Bombali virus and Reston virus, which only infect other animals like mice and bats. Ebola virus is a filamentous virus that has a curved, hooked, or spherical shape and is anywhere between 80 nanometers and 14,000 nanometers long. The virus appears to be thread-like and forms the shape of a 6 or a U in its curvature. The virus has an envelope and a glycoprotein surface that depletes the immune response of neutrophils. The glycoproteins project from the lipid bilayer surface and are 7 to 10 nanometers long. These glycoproteins are what allows the virus to attach to new host cells. Ebola virus attacks the body by targeting macrophages and dendritic cells. Glycoproteins from the virus bind to the dendritic cells in the body. The dendritic cells that are attacked can no longer signal T cells to fight off the virus. This inhibits our immune response and leads to fever and inflammation. The surface proteins of the virus bind to blood vessels, destroying cells and leading to hemorrhaging, or the bursting of blood vessels. Ebola virus contains 7 genes, including a nucleoprotein, glycoprotein, RNA polymerase, and the viral proteins VP35, 40, 30, and 24. The main role of the nucleoprotein is replication and transcription. Glycoproteins play a large role in binding and entering the host cell, as well as in membrane fusion. RNA polymerase is a key component in replication and translation. VP35 regulates RNA synthesis and keeps the virus from being recognized by immune receptors in the host. VP40 helps with transcription regulation, interactions with the plasma membrane, filament formation, and budding. It is the most plentiful viral protein. VP30 regulates and initiates transcription. VP24, lastly, aids in forming nucleocapsid and replicating the virus. Since Ebola is not functional without taking from other host cells, they use their extensive structure to replicate using a host cell. Ebola first attaches to the cell. The glycoproteins on the surface of the virus are used to bind to host cells. The VP35 protein stops the immune system from fighting off the virus upon attachment. Ebola virus then enters the cell through macropinocytosis. Glycoprotein 2 has a fusion loop that inserts into the membrane of the host cell and begins to fuse. From here, the RNA in the virus enters the cytoplasm of the host cell to begin replication. mRNA uses viral polymerase for transcription. Ribonucleoprotein is formed from the nucleoprotein of the virus and is sent to the host cell to translate proteins. Next, VP35 releases RNA from the ribonucleoprotein. VP40 and glycoprotein start the budding process. Then a new daughter cell is formed from budding and is released from the host cell to further infect the body. This is how the viral load increases after a small amount enters a host. Because of the disruption that Ebola has in the gastrointestinal tract and its ability to reduce the efficiency of the immune system, the microbiota is likely to become imbalanced after an Ebola virus infection. Pathogenic gram-negative bacteria have been found to enter the lamina propria of the infected person. In one study, 10% of previously infected people with Ebola experienced a bacterial infection. Ebola virus interacts with the human microbiota by creating an environment that allows for pathogenic bacterial growth. Ebola interacts with other organisms by infecting non-human primates as well. The virus has been found in bats, monkeys, gorilla, and chimpanzees. The environment of Central and Western Africa is more likely to allow for Ebola virus outbreaks because of the compact population, interaction with the animal environment, and an underdeveloped healthcare system. And here are my sources.
Hello, my name is Raquel, and for my project I chose the hepatitis B virus. What is hepatitis B? It is an infection that targets the liver and is caused by the hepatitis B virus. The hepatitis B virus is part of the Hepatinavirdae family and the genus Orthohepatinavirus. It is a spherical double-shelled structure surrounded by an envelope of a nucleocaspid with the HBsAg antigen that encodes the viral DNA. Hepatitis V affects both children and adults but can progressively get worse in children who got it passed from their mothers at birth. It is transmitted through infected bodily fluids such as semen, blood, and like I said, transmitted from mother to child at birth. There is acute or chronic hepatitis B. It is acute when sickness lasts around 3 to 4 months and the antigen disappears when liver function is recovered. It becomes chronic when liver inflammation lasts longer than 6 months. It has 4 phases, including the immune-tolerant phase, immune-active phase, immune control phase, and immune clearance or reactivation phase. In the immune-tolerant phase, viral load is high and there are no symptoms present. In the second phase, the virus replicates quickly and attacks hepatocytes. During this phase, fibrosis may occur as scar tissue develops, which can lead to cirrhosis or hardening of the liver. During the immune control phase, host immunity is high and can control viral load. So if you are in the acute part of this disease, your immune system will get you back to normal. However, in the immune clearance phase or reactivation, if your immune system is weak, which can be due to drugs or underlying conditions, viral load increases and reactivation occurs. The severity of the disease depends on what phase you are in and the interaction between immune response and viral activity. Symptoms of hepatitis B include fatigue, abdominal pain, loss of appetite, jaundice, dark urine, and joint pain. Those who are at higher risk should get tested regularly. These groups include pregnant women, those who were not born in the U.S. and did not receive the vaccine, infants born to mothers with HBV, men who have sex with men, people who inject drugs, and those with HIV. Immunization is the best prevention against this virus. The vaccine boosts our immune system, eliminating the surface antigen and controlling replication. Other ways to prevent hepatitis B are practicing safe sex, not using other people's personal belongings, and getting tested regularly. Thank you.
Human papillomavirus, HPV, is a virus that causes benign growths of the mucous membranes or the epithelium of the skin known as papillomas and commonly referred to as warts or verrucae. The virus is epitheliotropic, meaning it has an affinity for epithelium. Most commonly, the hands, feet, and anogenital region are sites for papilloma to form. Belonging to the Papillomaviridae family, there are over 170 strains of human papillomaviruses. HPV is the most common sexually transmitted infection. However, it can be transmitted sexually or non-sexually acquired. Some of the subtypes of HPV are more carcinogenic than others, meaning they have more potential to cause cancer, particularly genital and cervical cancers. Human papillomavirus is a small virus, approximately 55 nanometers in diameter, that has a circular structure contained in a naked polyhedron with 20 faces referred to as an icosahedral capsid. HPV contains a single molecule of single-stranded DNA, deoxyribonucleic acid. HPV is a non-enveloped virus, defined as lacking a lipid envelope. This so-called naked virus only requires its protein capsid and host detector proteins to infect host cells. Since the virus lacks an envelope, it provides itself with more resistance to extreme pH, heat, dryness, and simple disinfections, allowing it to be more virulent and typically causing host cell lysis. Papillomaviruses are an ancient group of viruses exquisitely adapted to their hosts in a tissue and species-restricted manner. The human papillomaviruses, HPVs, are responsible for significant morbidity and mortality in the form of various epithelial infections and cancers of skin, anogenital and oral sites. These high-risk human papillomavirus strains are frequently associated with invasive cervical cancers more than other types of HPV, indicating they are more carcinogenic than other HPV types. For instance, HPV-1 and HPV-4 typically cause warts on the feet referred to as plantar lesions, while HPV-2 typically causes common warts on the hands. Viruses themselves are generally metabolically inert. Human papillomavirus is no exception to this observation. The lack of independent metabolism is a result of the virus not possessing protein synthesis machinery and an enzyme system. HPV must rely on metabolic events within the cell to generate its component parts to replicate new viral copies. Infection essentially acts to change the cell's metabolic activity in order for the virus to replicate. The metabolism of HPV depends on the strain and the tissue it interacts with. For example, high-risk HPV, HRHPV, strains are associated with a greater change in host cell metabolism than less virulent strains that affect the cutaneous regions of the body. Human papillomavirus does not take in nutrients and grow to increase in biomass in the same manner as a bacterium or animal does when increasing nutrient uptake and subsequent size of the organism. Instead, it simply replicates by hijacking all of the machinery present within the host cell. The nutrients available to HPV, and the subsequent growth or replication of infection, depends on the strain and the tissue it interacts with. For instance, high-risk HPV strains are associated with a greater change in host cell metabolism than less virulent strains. So it can be inferred that more nutrients are available to these high-risk strains within these specific tissues and are able to grow at a greater rate. Human papillomavirus co-infections with other viruses can impact how the virus replicates and impacts the host. In a 2018 review of HPV co-infection with various other viruses observed that some viruses can be viral cofactors that contribute to HPV persistent infection. Epidemiological and experimental studies investigating human immunodeficiency virus, HIV, herpes simplex virus, HSV, 1 and 2, human cytomegalovirus, Epstein-Barr virus, BK virus, JC virus, and adeno-associated virus as viral cofactors in or therapeutic factors against the development of genital and oral HPV-associated carcinomas. Human papillomavirus infection can disrupt the natural symbiosis of healthy microbiota of various tissues on and in the body. The human microbiota plays a significant role in protecting the body from pathological infections. Environmental factors such as low standards of living, poor personal hygiene, diet, and smoking also impact the incidence and persistence of HPV infection. Smoking could influence the incidence and persistence of HPV infections by suppressing local immune function, increase cellular proliferation and turnover, upregulate pro-inflammatory factors, or cause host DNA damage resulting in increased susceptibility to infection. Thank you for watching and stay healthy, my friends.
Poliovirus. What is it? It's a human enterovirus that causes poliomyelitis. It's highly contagious and can be life-threatening. It usually targets young children, but adults are susceptible as well. It's spherical, containing a single-stranded positive sense RNA, a capsid, which is a protein coat, and no envelope. Poliovirus can be transmitted by fecal or oral transmission. This could be from contaminated food and water, or from droplets in the air from an infected individual who sneezes or coughs. The virus moves into the GI tract. This virus binds to the epithelial cells of the intestines and releases its RNA within the cell. In the cytoplasm, it uses the cell's RNA polymerase to make copies of itself and uses the ribosomes to make viral proteins. This production of viral proteins and RNA creates new polioviruses, causing lysis and releasing newly formed viruses into the interstitial fluid to be transported by lymph nodes to the bloodstream. From the blood vessels, poliovirus can move into the brain and spinal cord and eat away at the motor neurons. In more advanced cases, this causes muscle atrophy, paralysis, difficulty swallowing and speaking, and even death. There are 3 types of paralytic poliomyelitis: spinal, bulbar, and bulbospinal. Spinal poliomyelitis refers to asymmetrical paralysis, usually of the legs. Bulbar poliomyelitis affects the brainstem that controls the motor nerves that function in speaking and swallowing. In bulbospinal poliomyelitis, the brainstem is also affected and disrupts the proper sending of signals down the diaphragm, a muscle important in respiration, which can make it very hard to breathe. The first documented outbreak of poliovirus in the United States of America was in 1894. President Franklin D. Roosevelt contracted poliovirus as an infant and suffered from post-polio syndrome, which means that polio affected him many years after his initial infection. Roosevelt was 39 when he suffered paralysis in his lower limbs, which made him wheelchair-bound. He was one of the founders of the March of Dimes, which was originally named the National Foundation for Infantile Paralysis. This foundation supported poliovirus research and treatment. So, how do we avoid contracting this dangerous disease? Well, the best way is to receive the polio vaccine. In 1955, Dr. Jonas E. Salk created the first injectable inactive poliovirus vaccine. In 1961, Dr. Albert B. Sabin created an active oral vaccine which was administered through the mouth. Throughout the years, the polio vaccine was further developed to be more functional. In 1994, poliovirus was no longer a public health problem. Because of this vaccine, many countries, including North America, have eradicated this virus, although its impacts are still felt in some countries across the globe.